And good morning, everyone. Thank you all for attending. It is indeed my great pleasure to share with you our research on nanoparticles for cancer diagnosis and therapy. So our work focuses on developing, MRI and multifunctional nanoparticles for combat various types of cancers. So these nanoparticles are engineered to target tumors, overcome cellular barriers, enable non-invasive diagnosis. They serve as a carriers, delivering chemotherapeutic agents and genetic materials like DNA, siRNA, and mRNA, and immunotherapeutic agents directly to the cancer cells. So this approach allows us to monitor treatment efficacy and to detect residual stress disease, and by integrating chemo, radiation, and immuno and photothermal therapies, we aim to address the root causes of cancer, offering more effective and less toxic treatments. Today, I will highlight our nanoparticle-based therapies for brain and breast cancer. So despite advancement and cancer treatment faces significant challenges. The cancer burden remains enormous, with nearly 200 million projected deaths in the US in 2023, and a lifetime risk affecting 1 in 2 women and 1 in 3 men. Current treatments like chemotherapy and radiation are not specific, killing both healthy cells along with and cancerous cells, and leading to severe side effects. Drug resistance and cancer stem cell often prevent complete eradication, resulting high recurrence rate. For example, the brain cancer has a recurrence rate of nearly 100%, and breast cancer around 30%. Apparently, we need more targeted and effective therapies, and nanoparticles offer a promising avenue. Nanoparticles provide a versatile platform for cancer management, serving as a contrast agents for early detection and as a carriers for chemotherapy and biotherapeutic drugs. So they protect the drugs from degradation, enable targeted delivery to tumors with a controlled release, reduce the systemic toxicity, overcome drug resistance, and target the cancer stem cell. Theranostic nanoparticles and integrating diagnostics and therapeutic capabilities are particularly promising for detecting cancer-delivered drugs and monitoring treatment disease and effective delivered drugs. And the systemic injection of nanoparticle is the most effective cancer treatment approach. Once injected in blood, nanoparticle must bypass physiological barriers, maintain lung circulation to reach tumors. And surface coating and the size of nanoparticles can significantly influence their performance or the PK profile. Surface coating, like polyethylene glycol, enhance the stability, reduce the immune response, and prolong half-life. To avoid rapid clearance, nanoparticles should be sized between 10 to 100 nanometer. So large nanoparticles larger than 100 nanometer tend to accumulate in liver, while the smaller ones, less than 10 nanometer, are cleared by the kidneys. For treating brain tumors, nanoparticle must cross the blood-brain barrier. All at the tumor site, nanoparticle are needed to overcome intracellular barriers and be internalized by cancers to deliver effective therapies. So what are the common limitations of nanoparticles? So why nanoparticles offer significant potential for cancer therapy? Several challenges hinder their clinical translation. So they're easy to aggregate due to the high surface to volume ratio. They are difficult to maintain, small size, colloidal stability after drug loading. They're very difficult to load hydrophobic drugs, because of the physiological barrier. Mediums are the hydrophilic, difficult to pass the intracellular barriers, deliver a large amount of drug to tumor sites. Most of nanoparticles go to liver, stay there. If the nanoparticles are non-biodegradable, they can cause the toxicity. Also, and the complexity of multifunctional nanoparticle can compromise the clinical development. So our nanoparticles are engineered to address um, many of these mentioned challenges, providing biocompatible theranostic platform for cancer diagnosis and treatment. So they integrate multifunctional components, each such as targeting ligand and the drugs and the fluorophore. So our design is each component serve multiple functions to simplify our design. As illustrated in the figure, so our nanoparticles actually finally they can have a very good stability in the over the serum, so we can make a very uniform size, and they really show the high specific tumor targeting, and they can pass the blood-brain barrier. They are also allowed for the scalable and rapid production with the microfluidic devices. So our nanoparticle also demonstrate high therapeutic efficacy in various cancer models in mice and rats. And also, iron oxide-based nanoparticles show the non-toxicity and the immunogenicity in primates. Next, I will introduce one of the two major nanoparticle types developed in our lab, the biodegradable iron oxide nanoparticles with the superparamagnetic properties. So nanoparticles are detectable by MRI and have a wide range application in cancer research and therapy. They are synthesized. They can be synthesized using the aqueous co-precipitation and high temperature synthesis, so we can control the size of nanoparticle. So that we also control nanoparticles have a low macrophage uptake and a very low immunogenicity. So that we also needed to show the different coatings nanoparticles are stable in the serum. So alongside of iron oxide nanoparticle, we have developed the carbon and the graphene-based nanoparticles and to enhance hydrophobic drug delivery, offer unique benefits for cancer treatment. And these include mesoporous carbon and nanoshells for efficient drug delivery, magnetic responsive carriers for combined internal thermal and chemotherapy strategy for effectively cancer therapy, and the biocave nanoparticles that can increase the tumor uptake. We also developed the boron-doped graphene quantum dots as a contrast agents. So these contrast agents, a T1 contrast agent, have a long half-life and a safe profile. So we aim to use this contrast agents to replace current toxic half-life of gadolinium GDP, gadolinium clinical contrast agent. We now turn to critical application, iron oxide nanoparticle for brain tumor imaging and treatment. Glioblastoma, the most prevalent and aggressive primary brain cancer, has a poor prognosis. As shown in the survival statistics, brain cancer has a significantly lower 5-year survival rate compared to other cancers. Key challenges in treatment include it's a difficulty in different distinguishing the tumor cells from the healthy tissue. and also the vulnerability to surgical intervention and rapid growth and highly invasiveness. And also BBB restricted the access of the therapeutics, drug resistance. And brain tumor also has multiple cancers. It is very difficult to use each antibody to against brain tumor, each type of brain tumor. So our strategy focuses on the rational design of iron oxide nanotheranostics. Okay. So these nanoparticles feature magnetic core for MR imaging, biocompatible polymer coatings, and tumor targeting ligands, therapeutic payload, and BBB permeating molecules. So this multifunctional design enables simultaneous tumor imaging, target therapy, and treatment response monitoring. So by really carefully and selecting and combining these components, we develop very highly specific and effective nanotheranostic particles for brain tumor treatment. So the major hurdle in brain tumor therapy is delivering drugs across BBB while achieving tumor-specific targeting. So our nanoparticle address this through the strategic combination of nanoparticles. So we use the polyethylene glycol as a non-toxic polymer, facilitate BBB permeation. And chitosan is a biocompatible molecule. It has a large functional groups, allows the subsequent conjugation, and also enhance the stability of nanoparticle. And chitosan itself allowed to pass BBB. And so we use the cryotoxin and CDX. So the cryotoxin CTX is a scorpion and venom derived peptide, offers high specificity for brain tumor cells. And so it is reported that it really targets 95% of brain cancer types. And so cryotoxin also can penetrate BBB through the receptor-mediated absorption. So cryotoxin also inhibits the tumor evasion. So together, these nanoparticle chitosan PEG cryotoxin formulation enables effective brain tumor targeting and BBB penetration. And so effective tumor targeting remains a significant challenge despite the extensive research. So our nanoparticle, I'm going to show you a different type of nanoparticle that indeed to target the tumor cells as well as tumor tissues. So as shown here, these are the tumors, brain tumors. This is healthy tissue, healthy cells. So nanoparticle is red, and they have a high uptake by the tumors, not by healthy cells. This is also demonstrated MRI imaging of the tumors and showing black, and it doesn't show any contrast for the healthy cells. And so we also injected the control nanoparticles, nanoparticle PEG and nanoparticle PEG cryotoxin into the xenograft mouse model. And here's a tumor. So here's a tumor here. So we can see that compared to the control nanoparticles, nanoparticle PEG cryotox targeted nanoparticle mainly show in the tumor region in this model. We also demonstrate our nanoparticle targeted intracranial xenograft mice. So for example, that on the left shows the cryotoxin, the fluorescence labeled, and this row is that the GFP expressed the brain tumors. So we can see that fluorescence, the nanoparticle co-localize the brain tumors. So then we section the brain tumor, looking at the microscope, also the nanoparticles indeed also co-localize well with the GFP expressed tumors. And here's the MR imaging. So this is a pre-injection nanoparticle. We don't see anything and very little contrast. And after inject the nanoparticle 48 hours, we found that the MR imaging really showed the nanoparticle increase the contrast in the tumors regions, which confirmed by HE staining. We also use the transgenic mice and to verify the tumor targeting of our nanoparticles. So this is kind of this MRI show that nanoparticle increase. This is a quantitation. So the nanoparticle uptake by the tumors region, nanoparticle protoxin, they further increase over time. But for the control nanoparticle, they will uptake initially and then reduce. So the HE staining also show that nanoparticle protoxin and accumulate in the tumor regions, not the normal tissues. So after we really confirmed the targeting ability of our nanoparticles, and then we started to verify if our nanoparticle pass the BBB. So in general, we assess whether nanoparticle pass BBB through the like MRI, GDDTPA imaging, and Evans blue assay, or for those the animal model with intact BBB. Or we can check whether nanoparticle pass BBB using wild type mice. So this is a particular mice that has an intact BBB, the genetic engineer, the type of mice. So the EDTP basically looking at it to see if the BBB is intact. So we show that's intact, and we show the Evans Bruce assay basically will die every organs except the brain. So now we're ready to inject a nanoparticle. If a nanoparticle can show the specific targeting tumors, and we can say our nanoparticle passes BBB. Yeah. Okay, so this is a particular model. It's a medulloblastoma. It's not the glioma. Just doing the concept. So this is MRI imaging of the image of the mice, live mice with after prior and after injection of nanoparticles. So this is a target nanoparticle, this control nanoparticle. We have done many conditions. I'm just using this example. And the 40 hours post-injection, this is clearly we see that chlorotoxin nanoparticle and enhance the contrast of the cancers, not control nanoparticles. And so to visualize this, so we inject these nanoparticles labeled by 555 and the fluorescence. So inject these mice. So this is injected to the mice with the chlorotoxin nanoparticle, control nanoparticle, and a mice without nanoparticles. So we needed to, so this is a show that chlorotoxin only accumulate in the tumor regions. And so we needed to, so as you can see, we needed to shave the hair. Why? You can answer me. So I tell you the reason is this is basically that's this fluorescence, all the light, they have a penetration depth. So we have to shave their hair. So in order to see that fluorescence. So this is mice after 5 days. And after 5 days, then we sacrifice mice, then take out the brain. Because we know this brain, we wanted to see if exactly in the brain region. So these are the medulloblastoma. They occur in the cerebellum. So we found that the nanoparticle only accumulated the cerebellum, not the control nanoparticle, not the untreated mice. Okay. And so this is basically this results really show that our nanoparticle pass BBB, specifically and pass BBB. Okay. So now
you froze for a second, Miqin.
Yes. Can you hear me? Oh.
Yeah, you're back. You're back.
Okay. And
Just start on that slide.
okay, so it doesn't move. So after confirming our nanoparticle pass BBB, specifically targeting tumors, so I will present their application in brain tumor treatment. So the glioma remains the devastating disease with a median survival of only 12 to 18 months. And despite decades of research, progress in GBM treatment has been limited. Although the immunotherapies such as checkpoint inhibitors and T-cell therapy have achieved a significant clinical success in melanoma and hematological cancers over the past decade. But the checkpoint inhibitor immunotherapy have shown no clinical efficacy in recurrent GBM, so as shown here. And the standard treatment for GBM includes surgery, radiation, and temozolomide and chemotherapy. But the 2/3 of patients are resistant to the temozolomide due to high expression of MGMT DNA repair enzymes. So to address these challenges, we focus on delivering MGMT inhibitors using NP siRNAs to silence resistance genes. And we also kind of employ the tumor targeting nanoparticles to enhance radiation therapy. So our research has been actually focused on the improving current standard of care treatments with the goal of making our nanoparticles more feasible for FDA approval and clinical use, because the high cost of bringing a new drug to market remains a significant challenge. So the first studies, we use a conventional enhanced delivery of nanoparticle BG06. So basically, we use inhibitors for the MGMT inhibitors. So this is a MGMT inhibitors used in a clinical trial, but they are very toxic. So we try to use a nanoparticle conjugated with MGMT inhibitors and directly injected intratumorally. And we found that the nanoparticles that have a very minimum toxicity, they can extend and the mice survival of mice with the GBM. And so then next we try to use the nanoparticle for delivering siRNA MGT. So we wanted to improve that therapy because intratumoral injection is one approach. Can we use nanoparticle, systemic injection of nanoparticle? And so the next example is demonstrate nanoparticle-based therapy and siRNA for GBM treatment. So siRNA precisely silence aberrant genes with minimum dosing. So that siRNA is very specific. It's difficult to deliver large amount of drugs to the specific tumor site, but it is feasible to deliver a small amount of siRNA. So, however, siRNAs are very unstable, degradable, immunogenic alone. So our nanoparticle system ensures the stability, reducing the immunogenicity, and it supports repeated injections and facilitates by bypassing the various barriers. So this is like our nanoparticle is also same and has an iron oxide core and a chitosan and PEG. For siRNA delivery, we add one more layer with PEI or polyarginine. So those are the cationic polymers has a positive charge, so they can very effectively complex with negative charged siRNA. So we published a lot of paper on the nanoparticle siRNA delivery. It's here just kind of just do the demonstration. What usually use like nanoparticles, and we can be covalent bonding on the nanoparticles or just the complexing the siRNA onto the positive charged PEI, etc. And but our work, basically, we do first we check the in vitro study to make sure that nanoparticle can enter into the cells and escape endosome and release siRNA effectively. So we usually do the cells with the different type of cells and with the control nanoparticle and nanoparticle to see if they are uptake. So we label the cell with fluorescence, nanoparticles as a red color, and then we will do the gene expression studies to make sure that, so these are the GFP expressed cells, and then we use the nanoparticle silence GFP as a proof of the principle, right? So we will usually we also compare the commercial available other particles to compare the performance of our nanoparticle. So as you can see here, GFP expression, we can knock down very effective, just as our nanoparticle cortex can effectively knock down the siRNA. And then once we know they have very high efficiency for the siRNA silencing, and then we do the toxicity study. So make sure that our nanoparticle do not have any toxicity to the cells. So once we kind of worked out with all the siRNAs and some physical chemical characterization, the chemical composition, and the nanoparticle stability, so show the size, and then we will check the efficiency of nanoparticle in the vivo. So that we will first check is that kind of this is check the nanoparticle with the GFP express the brain tumors, and then we inject the nanoparticle that's silencing the GFP, and to see if we can once we can effectively silence the GFP, and then we do the survival study and measure the tumor volume reduction. So this is so this is like the GFP study. After GFP study, we delivered the therapeutic gene. So this is example that we delivered nanoparticle silencing MGMT. So that enzyme is a repair protein and cause a problem to make the TMZ, temozolomide, ineffective. So this is already show that our nanoparticle, the combined with siRNA MRT, combining with the temozolomide, it can really extend the survival as compared to the control untreated mice and the control temozolomide. So this is one is a temozolomide treated mice. Okay, so now I'll give you kind of a little bit of details that we also need to confirm that our nanoparticle indeed pass BBB. So this is what we usually do. We inject nanoparticle into the wild type mice. So after 10 minutes or 4 hours, or sometimes you can do 2 hours. So then we collect the tissue and label them with immune staining. So this is like a blood vessel, and the nanoparticles labeled by red. So we'll see if the nanoparticles actually in the blood vessel or it goes to the brain tissue. So for example, this is a 10 minutes, they actually goes to the brain tissue. So it's not in the blood vessel. So we know that they pass BBB. Okay, so if they do not pass B, we are not going to see any nanoparticle in brain tissues. So then we also do the toxicity studies into the all these very standard toxicity study that shows our nanoparticle do not have any toxicity. Okay, and in the so in a rat, and so then we actually also confirmed that we wanted to see if our nanoparticle really killed the cancer stem cell. So usually those are the very small cancer stem cell cause recurrence. So we use this GBM model. It's also PDX, patient-derived xenograft model. It's called GBM6 model. And that we really confirmed that they do have the stem cells. So that they showed all the stem cells markers. And then we also see that we collect the tissue, make sure that in the tissue, the nanoparticle really knock down the MGMT. And then we also showed the nanoparticle target the tumors. And then we also show that nanoparticle indeed that they cause the apoptosis of tumors, just using a typical assay. And then I also kind of want to show this is MRI imaging. This is a tumors, and this is after injection. This is initially see the big tumor black boundaries, and after 2 weeks injection of nanoparticle siRNA plus the temozolomide, the tumors disappeared. So this is, of course, not all the case. If all the case, we have the very good particle for treatment should go to the clinic right away, right? But we've seen actually quite a few mice like this. So it's really interesting, these particles. And then we kind of wanted to see if our nanoparticles can actually also, we apply this nanoparticle and deliver the genes we call against the APOL1, and that is a gene kind of for the the radiation resistance. So we wanted to inject this nanoparticle and combine with the radiation in a genetically engineered mice, which is provided by the Professor Erica Holland group. And so we basically found that we can show that our nanoparticles say similarly that can enter the cells efficiently and escape the endosome and can knock down APOL1 effectively, can also kill the tumor cells when they combine with the radiation. Okay, so we also apply. So once successful in the vitro, then we apply them in the vivo. So basically, we do the schedule usually is a tumor inoculation, then inject nanoparticle siRNA 1 day before the treatment, then collect the treatment 1 day after treatment. So give them enough time. So we also show that our nanoparticle design really show the extended survival compared to the other control nanoparticle or untreated mice or mice treated with the radiation only. Okay. Then, so I'm going to kind of tell you another interesting work is that we basically we can inject the nanoparticle cryotoxin into the tumors. This is a conventional radiation therapy, and we inject the nanoparticle cryotoxin only. So we found out that this nanoparticle cryotoxin only can amplify radiation. So this is basically produce more free radicals. And so this we, of course, in doing a bunch of the characterization, so we found that cryotoxin nanoparticle combination alone can reduce the tumors and extend the survival. And this is very interesting. It's because the nanoparticle cryotoxin without any drug. So it should be more easier for FDA approval. So if we inject a nanoparticle 1 day before the radiation, and that can really improve the therapy. And then we kind of study the injector nano to try to study the biodistribution and the safety in the monkeys. So basically, we wanted usually for the FDA approval, we needed to know the biodistribution safety of the nanoparticle. We can do biodistribution nanoparticle easily in rats, so we can kill the rats and study the biodistribution. But it's really challenging to get in the biodistribution study in monkey because monkey is too expensive. So this is a really exhibited advantage of iron oxide nanoparticles. So once we inject nanoparticles, we can check each organ. This is the liver, shows the nanoparticles kinetics basically, and nanoparticles increase and decrease, and in the blood, and so we can study the kinetics, and so we can study really about the distribution. of the nanoparticle in monkey. And then we also take the blood out and to study the toxicity of the nanoparticle to the blood and study the immunogenicity. So all the results really show that our nanoparticles are non-toxic and non-immunogenic. Okay. Here. So, and now I'm going to, so it's it, what so I'm going to kind of finish the nanoparticle therapy for brain tumors. So I'm going to shift my focus to the breast cancer. So the breast cancer is the most common cancer in women and the second leading cause of cancer deaths, including triple negative breast cancer, an aggressive subtype and metastasize to brain, lungs, and bones and the liver. So the TNBC treatments face challenges, lack of the targeted therapies, rapid progression and drug resistance and tumor heterogeneity and toxicity. So first line treatment included an anti-PDI and the chemotherapy, doxorubicin, and the paclitaxel. And we've developed a multiple nanoparticle formation for breast cancer with a key example highlighted. And so the first example is that we use a nanoparticle treating micrometastasis. So again, we use a nanoparticle with, this is an antibody. We are not talking about chlorotoxin anymore. And so we basically kind of found the nanoparticles as a small size, stable, and they can specific targeting the tumor region. And then we really demonstrated that after injection of nanoparticle, we can see where these nanoparticles can really target the metastasis. And this is a very early work, and I'm going to show you some of the recent work. And so we have, so this is a kind of interesting nanoparticle. So we developed the one nanoparticle and made of chitosome and the PEG and the paclitaxel. So as these materials, we do the self-assembled into kind of fiber. It's a long fiber with the diameter is with the height is about like 20 nanometer. The length is kind of be 1 micron. We can control that. And these particles is interesting when they inject in the blood, they break into 20 nanometer, 20 to 30 nanometer, very small. So as you know that it's very difficult to make the smaller nanoparticles. The smaller nanoparticle is very more efficient to get a tumor size. So that's the kind of motivation for us to design these nanoparticles. These nanoparticles fiber can break into, when they interact with the serum, they can break into very small size. So then we injected these nanoparticles in these triple negative breast cancer, very aggressive model. We found our nanoparticle can really inhibit or prevent the metastasis. With our nanoparticle, these tumors is going to further grow. And with our nanoparticle, and almost like 40 days, really stop growing. So, and also can really significantly reduce the tumor volume. We also, recently, we developed our nanoparticle for combined chemo and immunotherapy. So this is a kind of one example that I use a nanoparticle and with layer by layer. So we kind of, this is iron oxide nanoparticles so with negative charge. And then we place the triple negative, we found the particular ligand that can target the triple negative breast cancer. And then we coated with the doxorubicin positive charge. And then we also coat with the negative charge poly-IC. So this is a very compact, simple nanoparticles. And we found that these nanoparticles, very small size, they can really inhibit the metastasis, reduce the volume, and increase the survival. We also developed a lot of various carbon and graphene quantum dots for breast cancer, so that we can basically like magnetic responsible. So we inject nanoparticle. We remotely use the magnetic field to um, and remote control, let them heat it up. So then can once they get to the tumor size, then we um, control the release of drug, heat it up, release the drug. So they are very effective to kill all the tumors. So this is like a interesting. Basically, you can completely wipe out all the tumors. And this is like carbon-based shells that we can very simple, just one carbon base, the porous. We can uh, load a very high hydrophobic drug. So for example, the iron oxide nanoparticle, we usually do the chemo drug, is loading capacity is about 10 milligram. For these carbon-based ones, we can load it like 70%, 80% of hydrophobic drug. And they can be also very efficiently to reduce, eradicate the tumors. So recently, we also focused on a lot of more like a nano vaccines. So we injected a little bit early, so we'll be able to um, and actually really prevent the metastasis. And so next, I'm going to spend a few minutes on talking about how we develop nanoparticle for gene delivery in immunotherapy. So the gene delivery for immunotherapy involves delivering DNA and mRNA to tumors, release the cytokines to activate the immune cells, or directly targeted immune cells to the immune cell and kill the tumors. So the mRNA delivery, so this involves the mRNA delivery, DNA delivery, and yes, so the mRNA delivery is no insertional mutagenesis, no cell nuclear entry, rapidly transient expression. DNA is more stable than the mRNA sustained expression, insertional mutagenesis risk. So we now, as everyone is really trying to focus on mRNA, and because it's more safer, so we develop some nanoparticle for mRNA delivery. And so kind of give you a little bit background for the mRNA delivery. In general, there are two methods for mRNA delivery. So this is a viral carriers, very efficient for the mRNA delivery, but pose the risk of and in it's not safer and for virus. So currently, there's a lot of clinical success for the leading platform, and there's a lipids. Lipids, there's a multiple components. It's at least 4 or 5 components to assemble lipids. So this is inside the core is mRNA. But this is lipids is very labor intensive, harder to optimize the synthesis, and batch to batch inconsistency, clinical side effects, and a non-specific tissue accumulation requires a deep freezing for storage. So also difficult for the functionalization. So we developed a kind of polymeric nanoparticle delivery vehicle using very simple components. So we have one polymers which has a PFHA, just the 3 components, basically kind of mimic the lipids, but the lipids is made of the separate components. So we made one component with all the components and the compact mRNA, then allow this entire particles can efficiently get into the cells, release the mRNA, and do the therapeutic functions. So this is like a way, basically, is the process that allows us and the mass production, and we can control the size very well, and so the size very well. And we can actually really see the cell uptake well, and they can successfully release the mRNA. And we have our nanoparticles show the superior stability than the lipids and liposome. And so, for example, this is we see that our nanoparticle we can storage in the 15 days. We can still show the high transfection efficiency, but for the liposome, this usually the day 1, 0, and actually 7 days it starts to very degrade. A few days degrade, so they are not stable. So you have to really store them in a very very low temperature. So we have done a lot of in vitro studies, and so then kind of just to show a little bit of in vivo studies. And in vivo studies, once we successfully show the vitro studies, so we evaluate the nanoparticle in vivo. So first we'll do is inject the nanoparticle. This is untreated material, untreated mice, and these are the mice with the nanoparticle with GFP or luciferase, so we can see them if they actually really show the a nanoparticle can deliver the fluorescence as a proof of the concept. Okay, once that done, then we use nanoparticle carry therapeutic mRNA. So then, so the mRNA, this is experiments using the mice and injected with nanoparticle with interleukin mRNA, interleukin 12. So we show that this is a red is control nanoparticle. This the blue is our combo with the interleukin with the anti-PD-1. So the anti-PD, this is the green one is anti-PD-1. So that's the standard therapy using anti-PD-1. So we see the anti-PD-1 it doesn't really have a good effect in tumor reduction. So with the combination, we actually almost like really don't see much. So for example, here, initially it's big tumor. Once we inject the nanoparticles, and many of them don't show tumor anymore. And so we also check to see if kind of T cell accumulated in the tumor. It's interesting, a lot of T cell accumulated tumor, not in the other regions. And then we also study if they indeed induce a lot of the immune response. So we take the blood out to study if they release those cytokines. And then we also study the biocompatibility studies. And so make sure that the nanoparticles do not have any toxicity, immunogenicity. And so that once we kind of show the polymer is very effective and delivered mRNA, then we also apply the polymers on iron oxide nanoparticle, the same polymer coating, to see if that effective to deliver the vehicle. Because we love iron oxide because it's MRI detectable. So we found that our nanoparticles can effectively treat a variety of cancer, including breast cancer, liver cancer, and brain cancer. And we show the nanoparticle can effectively release mRNA and have also high and basically have a high transfection rate. So we did doing all the flow data show that they have an immune response. And so currently, so far, like our nanoparticle can transfect a variety of cells. Now we're working on the stem cells, like a neuron stem cell is very difficult. And also, can we transfect the immune cell itself? It's also very difficult. So recently, we use a nanoparticle to transfect the T cell and the macrophage and dendritic cells. So far, all these yes, these cells usually show very low transfection from the literature. They only show 10%, 20%. But so far, we already obtained a very high efficiency on all these three immune cells. Okay, so
Miqin, you want to leave some time for questions? Boy, that was an amazing talk, just an amazing talk. Questions from the audience?
I'd like to acknowledge all the dedicated team of the research and our sponsor who supported this work. Okay. Okay, question. I'm happy to answer your questions.
Anybody? think you silenced everybody.
Too much work.
Well, no, it was fabulous. Mike's, are you talking, Mike? No.
I was talking to somebody off screen.
Oh, okay.
And this is, I mean, I don't know that I can understand most of this stuff. I just listened and was impressed, but I think this is sort of the future, right? Is that eventually we want to put ourselves out of a job, and the way to do it is with this sort of thoughtful stuff and not the sort of primitive idea of cutting somebody's head open and physically removing tumor tissue. So, I mean, it's just great. I wish that, this got even more support than it does.
Yeah, one of the best parts of my career is collaborating with Miqin. So it's really been pretty fabulous. Miqin, thank you
Yeah,
very much. It was a tour de force. That was just amazing. And you did it on time with 5 minutes left. That was fantastic. You were worried about that. But thank you. All right. We're all everybody, and you know how to get a hold of Miqin. She's in the School of Engineering and she's in our department. She's part of our department, a professor in our department. So without further ado, thank you so much, Miqin. Thanks for getting up early and doing this for us. And
thank you.
and for the students and residents that want to work with Miqin, please reach out. You don't have to reach out to me. I mean, I certainly can put you in touch with them, but her labs are rocking. They are busy labs with lots of students. She takes on all kinds of students. She's taken on R25 summer students. She takes on postdocs. She takes on she loves neurosurgical residents. So thank you, Miqin.
Yeah, I just want to say something that I think Rich and I have in a training grant. We trained quite a few residents. I think everyone got the faculty job, right? It's in a very good university. Somehow, yeah. So I was kind of busy for writing my training grants. Some point I needed to do that. And at the time, we actually our project can really support almost like more than 10 doctoral students and postdoc fellows each year. Because my department, I think the administration is not great. Maybe I should do the neurological surgery because it's too much work for me to do the training grant. And by the way, so this is I think is and Richard, this is like I take a few minutes because the work for the nanoparticle for brain tumor targeted is really solid. So I think I just need someone who actually work on the clinical trials. And kind of these are already ready to bring them to the clinical. I'm just kind of too busy. And also, in addition to the nanoparticles, so my group is really doing all the biomaterials you can imagine, like injectable gels. We do injectable gel for the knee repair, for the disc repair, for the spinal cord. Recently, we collaborate with Christopher and for the spinal cord and repairing. And we do like my lab also do a lot of stem cells. So we do a lot of human-induced pluripotent stem cells, and we differentiate them into the like cortical neurons. So basically, we develop the 3D scaffold that is more efficient for the stem cell renewal or stem cell differentiation. We create a lot of different models. So we do like a brain organoids, and as well as brain tumor spheroids for the drug screening. So we're yeah really
Very versatile nanoparticle.
yeah, it's very interesting, all these brain tumors. So uh, anyway, I know I need a more actively collaborating with the professor in neurological surgery. So sometimes I get too busy, then I get something interested, and it just it cannot stop. But we do have all the materials available. Rich, that I think some of the faculty, your faculty also reach me. They kind of interested in transfected 293 the T cell stuff like that. We have all the materials actually. It can really have all the transfection. So at some point I probably needed to write some grants that can make materials that allow me to provide them to other people. So right now we're kind of in a lack of the resources to produce all the material to send it to the collaborators, right? That's kind of prevent a little bit of collaboration. Otherwise, yeah, we have a really good materials worked out very thoroughly. Mm-hmm.